You're listening to music from Smithsonian Folkways.
Good afternoon. Thank you for joining us and your fellow friends of the Smithsonian members for a conversation today about the Smithsonian Initiative Movement of Life. I am Colleen Moore, Assistant Director with Friends of the Smithsonian. And before our program gets started, I want to take a moment to say a sincere thank you for your steadfast support this past year. Your dedication and enduring commitment helps to continue our research into species and ecosystems across the planet, like the collaborative project we will learn more about today. Smithsonian museums and the zoo are working to reopen safely, but while we are closed, we are still producing exhibitions, sharing the Smithsonian's vast resources online, and continuing our groundbreaking research across the globe. Your continued giving in 2021 supports this ongoing work. Thank you for being there for us year after year. A few quick notes before we get started. We will be hosting a question and answer session at the conclusion of the presentation, and we encourage you to send your questions in during the program using the Q&A feature at the bottom of your screen. We are also providing closed captioning for today's event. We will hear from 3 speakers today. Each one will be sharing their screen individually. There may be a short pause while we transition from speaker to speaker. And now I'm pleased to share with you more about Movement of Life and to introduce our panelists for today. This initiative takes advantage of the breadth and knowledge across the Smithsonian, bringing together scientists to understand key aspect in preserving biodiversity, how all living things move across the world and interact with their ecosystems. Our speakers are from 2 of the units involved in this program, the Smithsonian Environmental Research Center and the National Zoo and Smithsonian Conservation Biology Institute. Today, we will focus on land, sea, and air movement. And if your interest is piqued, the Movement of Life will also be hosting a program on Earth Day. We will give you more information on how you can attend that event at the conclusion of today's program. And I found a quote on the project's website that I feel most eloquently describes this project. All movement, from daily forays of bees to the transcontinental migration of birds, is critical to life on Earth. Smithsonian's Movement of Life initiative aims to advance the understanding of how all living things, big and small, move across changing land and seascapes to better sustain a biodiverse planet. Our expert panelists joining us today are Amy Scarpinato, Matt Ogburn, and Jared Staubach. Amy Scarpinato is a research ecologist and conservation bird specialist at the Smithsonian National Zoo and Conservation Biology Institute Migratory Bird Center, which is dedicated to understanding, conserving, and championing the grand phenomenon of bird migration. Before joining the Smithsonian, Amy worked as an itinerant field biologist, working on nearly 15 different research projects with many different species of songbirds, raptors, waterfowl, and shorebirds. She has always had a passion and curiosity for science, nature, and unraveling mysteries, such as why, how, and where birds go. Matt Ogburn is a marine ecologist leading the Fisheries Conservation Lab at the Smithsonian Environmental Research Center. His research focuses on providing the best scientific information available to inform fisheries management and conservation decision-making from local communities to global institutions. Much of his research is conducted in the Mid-Atlantic region, including the Chesapeake Bay, addressing topics from oyster restoration and dam removal to sustainable management of fisheries for blue crabs, finfish, sharks, and rays. And Jared Staubach is an ecologist at the Smithsonian Conservation Biology Institute's Conservation Ecology Center and the program coordinator for the Smithsonian's Movement of Life initiative. His research focuses on the factors that affect the abundance, distribution, and movement patterns of large terrestrial mammals, incorporating emerging technologies such as unmanned aerial systems, GPS tracking collars, and high-resolution imagery to better understand and monitor changes across the planet. One of many collaborative teams that Jared has been on include the successful reintroduction of the scimitar horned oryx to the wild. And now I'm pleased to turn the program over to my colleagues.
Thank you, Colleen. Hi, my name is Amy Scarpinato. I'm a research ecologist and bird conservation specialist at the Smithsonian Migratory Bird Center and I'm excited to kick off this event today and tell you about our project tracking the National Zoo's black-crowned night herons. Oops, there we go. As I mentioned, I work at the Migratory Bird Center, which was founded in 1991 by an act of Congress, incorporated in 1997 as part of the National Zoo, and came under the Smithsonian Conservation Biology Institute in 2010, where it remains today. The Migratory Bird Center is dedicated to understanding, conserving, and championing the grand phenomenon of bird migration and seeks to clarify why migratory bird populations are declining through an understanding of bird biology. I primarily work on a project called the Migratory Connectivity Project. As advancements in technology and the ability to track a greater number of species increased, the Migratory Connectivity Project was created in 2009 to facilitate development and testing of new technology and act as a portal to gather and disseminate information. We aim to advance the conservation and understanding of animals throughout their full life cycle by promoting the science of migratory connectivity. Migratory connectivity is the linking of populations between one life cycle stage, such as breeding, wintering, or migration, to another life cycle stage. In the graphic on the right, we can see from top to bottom examples of high, moderate, and weak connectivity. With high connectivity, we can see there's separation among the populations on both the breeding and the wintering grounds, and for weak connectivity, a high degree of mixing of breeding populations on the wintering grounds. Migratory connectivity is key to conservation action and species survival. We need to understand where birds go to allow for more effective investment and informed decision-making so we can mitigate impacts at the appropriate place and time. To answer these important questions about migratory connectivity and full annual cycle biology, in the last 5 years, the Migratory Connectivity Project has worked with over 50 different organizations, deployed close to 1,000 tags, and tracked over 25 species, whose tracks are represented by the different colors on this map. In addition to our tracking program, one of our main projects is Discovering Unknown Migrations, the Atlas of Migratory Connectivity for the Birds of North America. The atlas will be published in fall 2022 by Princeton University Press, and the atlas synthesizes all available connectivity data, including bird banding, genetics, isotopes, and tracking data to help us gain an understanding of migratory connectivity for North American migrants. Additionally, this synthesis allows us to identify gaps in information. And it was this idea of discovering an unknown migration and filling in the gaps in information for migratory species that prompted Smithsonian scientist Paul Bartsch to initiate the first systematic and scientific bird banding in North America when he banded 23 black-crowned night herons with a small metal band that read, "Return to Smithsonian Institution," with the year and a serial number. Over the next couple years, Bartsch banded about 100 black-crowned night herons. While he hoped to understand more about the migration routes and wintering locations of individuals in the breeding colony, the only recoveries occurred locally a few months after banding and therefore provided no information about the question of where they wintered. So now I'd like to introduce you to this bird that captivated Paul Bartsch and was the first official species banded in North America, the black-crowned night heron. As it's so aptly named, it is most active at dusk or night. It feeds in mainly aquatic habitats such as wetlands, lakes, or streams, and nests or roosts in trees. They are colonial nesters and opportunistic feeders, meaning they have a varied diet consisting of small fish, crustaceans, reptiles, small mammals, and aquatic plants. It is the most widespread heron in the world, and in most areas, populations are considered stable. However, in 10 states, black-crowned night herons are listed as endangered, threatened, or of conservation concern. So now that you know a bit more about the herons, let's revisit the colony at the National Zoo. Following Paul Bartsch's banding study, the black-crowned night heron colony continued to capture the attention of many over the next few decades, yet the mystery of where these birds go when they leave the zoo remained unsolved. Today, the colony still breeds in the same trees around the birdhouse and is estimated to have about 100 breeding pairs. Scouts arrive in late February, early March to check out the nesting colony and the trees around the colony. and a few weeks later, the colony begins arriving in large numbers. Chicks hatch in April and May, and the birds depart the zoo in late August, September. So we know the herons are at the zoo from March to August, but as Paul Barch asked, where are they the rest of the year? Over 100 years later, technological advances in animal tracking are facilitating the proper investigation of Barch's questions about the migratory routes and wintering locations of black-crowned night herons, as well as many other migratory species, because tracking makes it possible to measure individual movements throughout the annual cycle, even when animals are in remote locations. However, the greatest challenge to tracking migratory animals is reducing the size of transmitters while increasing the accuracy and amount of locations. Often a trade-off exists between the weight of the bird and the weight of a transmitter, its battery life, accuracy and number of locations, and how the researcher receives the data. The Black-crowned night heron tracking project is an example of how we at the Migratory Bird Center are pushing the field of animal tracking technology to understand and conserve migratory birds. In this study, we use 3 types of transmitters, the first 2 being battery-powered satellite transmitter and solar-powered satellite transmitters. Battery-powered transmitters last about 1 year, while solar-powered can last up to 5 years. So both types transmit locations to satellites passing overhead, which are then relayed to a receiving and processing center where the data is processed and sent to my computer to provide the coordinates of the bird's locations. This process allows researchers to track a species in almost real time. In 2015, for the first time, we deployed solar-powered GPS GSM or cell phone transmitters on black-crowned night herons. Cell phone transmitters collect the GPS location of a bird every hour and transmit locations via the cellular network every few days. Basically, I would get a text from a bird telling me it had just transmitted data. The cell phone transmitters provide more accurate locations than the satellites we used, but there can be issues with the coverage of the cellular network or weak signals, the "can you hear me now" problem in some areas that can prevent the data from being transmitted. Additionally, the herons are most active at dawn and dusk, so it was a mystery to see if the solar-powered battery for both the satellite and cell phone transmitters would remain at high enough power levels to collect and transmit their locations. Now that we have the technology, it's time to figure out where these herons go. In 2013, we began a collaborative project between the Migratory Bird Center and the zoo's birdhouse staff. We captured black-crowned night herons by baiting them into flight pens. They were then weighed, banded with a unique identifying bird band and color band, and received a transmitter. Following animal safety and welfare guidelines, the transmitter is attached to the back of the bird with a harness as if the bird's wearing a backpack. After attaching the transmitter and checking for proper fit, the bird is released. In total, we deployed 18 tags, 12 satellite and 6 cell phone from 2013 to 2018 that successfully transmitted wintering locations for 13 birds. We tracked herons throughout the DC metropolitan region, as well as to North Carolina, Florida, Cuba, and Nicaragua. Birds wintered in many different habitats, including agricultural fields, rivers, a golf course in Florida, and national parks in the Florida Everglades and Cuba. We tracked most birds for about a year, but 3 birds for more than 2 years. These birds all return to the zoo to breed every year and also use the same wintering areas each year. In cases where transmitters stopped working, we were still able to see birds around the zoo colony. In fact, the bird that flew to Nicaragua, its transmitter stopped transmitting in 2019, but last summer, it appeared at the zoo and was seen by one of the keepers. Additionally, we were able to identify the heron's local and post-breeding movements. We can see that herons breeding at the zoo frequent the Potomac River and Rock Creek daily during the breeding season. After breeding is complete, many herons disperse to lakes and rivers throughout the DC region before beginning their southward migration. This also highlights the importance of taking care of our urban parks, streams, rivers, and wetlands. We just had a paper accepted highlighting the tracking data and wintering locations in this project. And our next steps are to investigate migration routes and winter habitat use and diet. And we could also ask questions about the colonies, such as what percentage of birds born in the colony return the following or subsequent years? What is the breeding success rate of the nests and fledglings from the colony? We could compare the migration distance and location of adults with their breeding success at the zoo. In other words, we may have solved Paul Bartsch's mystery, but we created many more of our own. So if you'd like to learn more about this project or the Migratory Bird Center and Migratory Connectivity Project in general, please visit us at the newly renovated birdhouse opening hopefully next year. Here you'll be able to also see the heron breeding colony up close and in person. If you can't join us in person, you can watch a great video of the whole heron banding process on the Smithsonian Channel YouTube. Last week, Smithsonian Side Door podcast released an episode featuring this project called Hot Bird Summer. And you can also read expedition blogs about this project and all of our other research on the Migratory Bird Center website, and follow us on social media. I'd like to say a big thank you to all of the people that made this project possible. Thank you to all of you for listening today and supporting the Smithsonian and our research. And with that, I'll pass it over to Matt Ogburn, who will take us from the skies to the seas.
Thanks, Amy. I'm excited about this opportunity to share some of the work we're doing at the Smithsonian Environmental Research Center, tracking species underwater. The goal of the work that my lab is doing, the Fisheries Conservation Lab, is to investigate the ecology, management, and conservation of marine and estuarine fisheries and ecosystems. But one of the unique challenges that we have in working in the oceans and in coastal estuaries and rivers is that our species are hanging out underwater. So they're in a place where they're harder for us to observe as humans because we don't spend a lot of time in the water. But they're also species that we can't really track with a lot of the same technologies as you'll hear about from Amy and from Jared, because the satellite technology that they're using only works when animals are at the surface. So you can use it to track things like whales or sea turtles and a few species of sharks and others. But for most of the species that live in the ocean and in our coastal bays and estuaries, we have to use other ways of tracking them to be able to understand how the individual animals are moving around. So we're using sort of different technologies to try to answer a lot of the same kinds of questions about what animals are doing and how can we better conserve and manage them. I'm going to give you 3 different examples of some of the work that we're doing that highlights both different species we're studying and different types of management questions and conservation questions, but also 3 different types of technologies. The first species I'll talk about is the blue crab, Calonectes sapidus, which if you live on the east coast of the US or the Gulf Coast somewhere and love eating crab cakes, this is probably the crab that you're eating. And blue crabs actually have a really interesting history with the Smithsonian. Their scientific name, Calonectes sapidus, which means beautiful swimmers, was actually named by a Smithsonian curator named Mary Rathbun, who worked at Smithsonian from 1884 to 1943 and described over 1,000 different species of marine animals. And then later, there was a book called Beautiful Swimmers, which is an excellent story of the blue crab fisheries and the blue crab itself in Chesapeake Bay that was written by William Warner. That's a Pulitzer Prize winning book. So we have a long history at Smithsonian of thinking about this species and working on it. And the work that I'm doing follows on work that CERC's director, Tuck Hines, started when he came to Smithsonian in the 1970s. But one of the reasons we're working a lot on blue crabs is because they're one of the most important fisheries in Chesapeake Bay and in the US. And there's a period of time, especially from the 1990s into the 2000s, when the population of crabs was really low and the fishery itself was actually declared a disaster, which allowed federal funds to flow to help fishermen and to help scientists learn how to recover the fishery. And what I'm showing in this graph here is date across the bottom and number of spawning age female crabs. So these are reproductive female crabs in Chesapeake Bay. And that number of female crabs had fluctuated a lot over history. The fishermen here talk about that a lot. It changes a lot from year to year. But what was really unusual in the 1990s and 2000s is that the numbers of females dropped to a low level and stayed low for a long time. And one of the lines of research that helped figure out what was wrong was work done at CERC that's very similar to the bird banding that Amy talked about, where we put these tags on the backs of the crabs. You can see in the photo here, it's a similar thing. We offered a $5 reward to anyone who caught a crab and called in the number or reported it on a website. And what we discovered from years of tracking these female crabs is that females from all over Chesapeake Bay would mate, and they actually only mate once in their life. And so that mating is really important. And then the females only, not the males, migrate from further up in the bay, sort of following the arrow drawn on the map here, down to the saltier lower part of the bay to release their eggs. And that allowed scientists to provide guidance to fishery managers to close a large portion of that lower part of the bay where they're reproducing during the summer, during the reproductive part of the year, close it to fishing. And since that and several other regulations were put into place in 2008, the population bounced back really quickly and hasn't been great every year, but has been increasing and decreasing, fluctuating like it used to in the past. So we think this research has been really critical for helping design the management that's helped recover the blue crab fishery here in Chesapeake Bay. The second species I want to talk about is a group of two species that collectively are called river herring. The one shown here is an alewife, and there's one that looks almost exactly like this called a blueback herring. And you might wonder why I'm showing pictures of rivers here when I'm talking about ocean animals. But these are fish that are migratory fish that behave a lot like salmon, where they live most of their life in the ocean, but they're born in freshwater streams. And then when they're old enough to reproduce at about age 2 or 3 or 4, they migrate back from the ocean to those same streams to reproduce and create the next generation of fish. And the populations of these fish declined dramatically in the 1950s to 1970s because of overfishing, too much fishing for them in the oceans. But the other main impact on the population was that large amounts of the habitat where they reproduce in streams and rivers is blocked by things like dams, like this one shown here. This dam is Bloody Dam, which used to be on the Patapsco River, which is located right next to Baltimore-Washington International Airport here in Maryland. And when these fish would come in from the ocean to reproduce, they would block this dam, be blocked by the dam, which is about 3 stories tall, and not be able to move further upstream. The photo in the upper right is what it looks like since 2019. This dam was just removed recently to allow the fish to move back into their habitat upstream. And the research that we're doing is trying to track the fish to see if they're actually using that habitat upstream, how much they're using it, and how often they're using it. And we use a different type of tracking technology to do that, which you're probably really familiar with. So the picture at the top left here shows us tagging, inserting this tiny little tag into the fish. And that tag is the same technology that your dog or cat might have in it as a microchip. When you go to the vet and they wand your cat or dog, it beeps and gives them an ID number that helps them make sure that your dog or cat can be tracked if it's lost. We use the exact same thing for these fish. And then the way that we track them, instead of catching them and wanding them, which we can do, we put these antennas out in the river that are really long, that basically work like easy pass for fish. You know, if you're driving on the highway and get an easy pass toll, this is the same basic technology where we can track fish that swim over these easy passes for fish that we put out in the river. And this year is the first year we're able to track them to see if they're moving upstream past the dam. We haven't seen one do it yet, but it could happen any day now. So we're really excited to see this year if we can track these fish moving in from the ocean and moving into the habitat upstream of the dam that's been closed to them for over 100 years. And there's also a Side Door podcast story that I believe is coming out next week about this work. And so you can look out for that in the near future. And the third type of technology and type of species I want to talk about is coastal sharks and stingrays. So for these guys that are out there swimming in the open ocean, we can't use that same easy pass technology either very easily. So we use a kind of technology called acoustic telemetry, where we put a tag on the animal or in the animal that sends out a series of pings or clicks that encode a unique number, so that if we put a receiver in the water, and I'll have actual pictures of them in the next slide, the tags and receivers, if they swim near that receiver, that series of pings or clicks will get decoded by that receiver and tell us what animal was detected there and when it was detected there. And because sound is transmitted a lot further in the water than it is in air, we can hear these from a half mile to even a mile away from the receiver. So this is what some of the receivers and tags look like. And the different tag sizes are just for different size animals. And the map on the right here shows the Atlantic Cooperative Telemetry Network in green, the Ocean Tracking Network in blue, and the FACT network in red. And each dot is a location where one of these receivers is out there in the ocean or along the coast listening for these fish. If they swim near any of them, we get that information back from researchers working in that area. So we're at Smithsonian leading this sort of mid-Atlantic and Northeast US network that helps researchers share this data. And here's one of the projects that we're using this kind of technology for. The dusky shark is one of the shark species that's most endangered in the US. A lot of species of sharks on the east coast of the US were overfished up until the 1980s or so. And since then, US shark fisheries management has been one of the most sustainable shark fisheries in the world. But this particular species matures at a pretty late age and doesn't reproduce as fast as others. So it's been a lot slower to recover. And it's accidentally caught by fishermen that are out trying to catch other species, recreational fishermen, sort of like what's shown in the picture on the left here. This is actually a researcher that's out with a recreational fisherman trying to catch sharks for us to tag. But if you're out trying to catch black-tip sharks off the East Coast, you might encounter one of these dusky sharks. So we're using the data that we get from these tags, which are shown in the gray circles here, to be able to understand where these sharks live during different times of the year. So each gray dot is a place where one of these sharks has been detected on a receiver that's out in the water. And they move north in the summer off of Long Island, and then south to North Carolina during the winter. And from a conservation perspective, there are two particular things we're looking at. One is the sort of purple magenta colored area is an area that's closed to shark fishing in the winter to help protect this species and a few other species. And so far what we've seen is that in general, that purple area is protecting them pretty well through most of the winter, but the sharks actually show up there about a month before the no fishing zone takes place or goes into effect. So we're hoping to work with managers to adjust the timing of that closed area a little bit to help better protect this species. And then the other thing that you can see in the inset part of the map that shows the yellow sort of squares, that's a wind farm lease area. The East Coast is about to have a lot of wind farms built, and we're tracking these sharks and other species to try to understand how they might interact. Could be positive, could be negative with those developing wind farm arrays out in the ocean. And we'll be providing this data to the federal government to help make that assessment. I hope you've enjoyed hearing a little bit about some of the species that we're working on in the water, and I'll hand it over to Jared to talk about some of the work he's doing on land animals.
Thanks, Matt. Yeah, what I like to do now is essentially build off of what Amy and Matt described, but instead of focusing on avian or aquatic species, I'll draw your attention to some terrestrial mammal studies that we have ongoing at the Smithsonian. Before doing so, first the thing I want to do is to show an animation which highlights the incredible phenomenon that we refer to as migration. Harold, could you play that video? So here, we're following the long distance movements of white storks as they move around the Mediterranean Sea and then across the Sahara Desert to wintering grounds in Southern Africa. Along the way, the storks stop over in East Africa and they overlap with some wildebeest that we are currently tracking before moving further to the south, all the way down to South Africa, where again, they overlap with some other species that are being tracked, like buffalo, and then with elephant. The storks then make this remarkable reverse journey, traveling thousands of miles back to Central Europe, completing this annual cycle. This animation highlights that movement is important and that we all live in an incredibly connected world. All species move to survive, to avoid predators, and also to reproduce. Terrestrial mammals, however, are at some of the highest risk of extinction simply because they share the same landscapes that we do. Roads, pipelines, railways, and in this case, fences, all pose significant barriers to animals trying to acquire the resources they need for survival. As a Smithsonian scientist, we don't simply want to be part of a team that tracks and monitors species or ecosystem extinction. Instead, and speaking for both Matt and Amy, we want to be part of the solution. So in the next few slides, I'm going to showcase a few of the projects that are currently ongoing at the Smithsonian. In each case, tracking movement is an essential aspect to either evaluate reintroduction success, to understand space use requirements, or to respond to potential threats. One of the first projects that I worked on when I came to the Smithsonian was the reintroduction of scimitar horned oryx. Once widespread across Sahelian grassland areas in North Central Africa, this species went extinct in the wild in the 1980s. In 2016, I'm happy to say that Smithsonian was part of a large international team to introduce individuals of the species that have been bred in captivity for the past 30, 40, or even 50 years. These animals were reintroduced to a large protected area, the Wadi Rime, Wadi Atchiam Wildlife Reserve in Central Chad. Over the past 5 years, we've reintroduced approximately 170 adults, and we currently have over 200 adults on the ground in Central Chad. We certainly have had some setbacks, but the important thing to highlight is that we're incredibly optimistic about the future of this species, and we've also had greater than 80 calves born in the wild. This indicates that animals are adjusting to this novel environment. In addition to the species, we're also working with local people. So you can imagine someone for the first time in their lives, like this young adult, seeing a scimitar horned oryx for the very first time and the excitement. In Southeast Asia, our scientists have been tracking elephants for over a decade. Scientific goals of this project are to understand how these majestic mega herbivores move between dense forest patches and between human-dominated agricultural land. In 2018, we were unfortunately alerted to an emerging poaching crisis. Instead of being poached for their tusks, however, like their African cousins, these Asian elephants were being poached for their skin. This essentially meant that any animal was being targeted, whether they were a male or a female, young, adult, or even infant. As a response, we drew attention to what was going on in the region. First, publishing a scientific article to draw international attention, but then also working with local officials across the region, we were able to increase the ranger patrol effort, essentially reducing the poaching threat across the region. Since that time, we haven't had any record of animals that were poached. In East Africa, I'm part of a team that's been working on white-bearded wildebeest for the past 10 years. Wildebeest are the dominant herbivore across grassland systems in East and Southern Africa, affecting nearly every aspect of the ecosystem. Wildebeest, however, are also incredibly sensitive to human disturbance, and they're also extremely bad jumpers. So here, this small group of wildebeest is running directly towards what is actually a pretty minor fence, but these animals will have trouble and they are unable to track the resources that they need to survive. So what we've been doing is we've been working with local teams in Kenya. I've zoomed into an area in Southeast Kenya, which is the northern part of the Serengeti Mara ecosystem. The figure, what it shows is all these gray dots. These dots are all individual wildebeest that we tracked across this system. And what it should give you is an assessment of the distribution of these animals and also the density of animals on this landscape. So particular areas that are used more often than others. Wildebeest across this region move from the western part of the ecosystem, the Mara Plains, to the eastern part of the ecosystem, to the Loida Plains. So movement is essential for their full life cycle biology. Also on this image, on the eastern side, on the right side of your image, what I'm showcasing is change that we're observing across the landscape. So because we have satellites of this area, we've been able to go back in time and catalog which areas have been converted or at least have fences within these pixels. So what you see in 2011 is that fencing was rather restricted. It was just basically one large area in the northeastern part of the ecosystem, roughly 450 square kilometers. Just 10 years later, fencing has quadrupled across this system. Roughly 2,000 square kilometers of fencing now exist. What we're essentially doing is putting those two data sets together. So where did animals historically move across the area and what areas have been fenced? In doing so, we can prioritize conservation action. And right now we're working on providing this information to spatial planners across the region with the ultimate goal of protecting this fragile ecosystem for future generations. And lastly, I want to draw your attention to a large collaborative project that we're working together with the Giraffe Conservation Foundation. Right now, we're establishing a giraffe science initiative across the entire continent of Africa to address the problem of rapid decline in giraffe. Now recognized as 4 distinct species of giraffe, most people are unaware that only 111,000 individual giraffe now exist across Africa. Part of what we're trying to do is simply understand how much space these individuals need throughout their lifetime. And in other cases, we might be taking extreme measures, like this picture shows, of actually translocating animals from one location to another. So taking them from a place of abundance and translocating them to another place where perhaps they may have been extirpated 10, 20, or even 50 years ago. You can imagine this is a difficult process. These are 19 to 20 foot tall animals and they weigh 2,000 or sometimes even 4,000 pounds. With that, and on behalf of Amy and Matt, I just want to thank you all for your time and attention. If you're interested in learning more about these programs or others, please visit our website and join us as we do our very best to manage and protect a more biodiverse planet. Thank you.
Wonderful. Thank you so much for sharing your work with us today, Amy, Matt, and Jared. Good afternoon to all. I'm Deborah Heller with the Friends of the Smithsonian, and I hope you enjoyed the presentation today and learning more about the work Smithsonian is doing to protect and preserve species and ecosystems. I have the privilege of asking our panel some of the questions you sent in. We will answer as many questions as we have time to in the next 15 minutes or so, and you can continue to send them in as they occur to you. So just to kick us off, we've got a question for you, Amy. Has the construction of the new birdhouse and other disruptive activities at the zoo affected the heron population and its numbers, reproduction, or other behavior?
Yeah, this is a great question. Thank you. And awesome job, Matt and Jared. I want to say, enjoyed your presentations. Yeah, so the daily activities at the zoo have not been shown to affect the colony. We don't have a systematic nest count protocol in place, but the birdhouse keepers have been keeping track of the number of nests over the last 10 years, I think, and it's remained stable, about 100 breeding pairs. Unfortunately, last year, due to COVID, we weren't on the zoo grounds, at least myself wasn't able to, so we weren't able to get a look at the nest counts. From the birds we had tagged last year, we had 2 birds, and they were at the zoo and on the Potomac River. So that's all I really can tell from that. I did get an email from one of the birdhouse keepers 2 weeks ago, and she said about 30 of the scouts for the colony have returned this year. And we have 1 bird that's left tagged from our whole group. And that bird was still in Florida at the same time at its wintering grounds. So hopefully in the next few weeks, since the scouts came in and checked it out, we'll be able to look at what the colony is doing this year. And now that hopefully we can return to normalcy. But yeah, great question. Thank you.
Great. So Matt, one for you. What happens to the female blue crabs after they release their eggs? Do they die? Or is one mating event good for all future spawning?
Yeah, that's a really great question. Thanks for that question. So the blue crabs have sort of a really interesting, unusual to us reproductive cycle. So they mate once, but the females are actually able to store that sperm for the rest of their life, which is a couple more years, and then fertilize multiple batches of eggs from it. So what we think happens for most females in Chesapeake Bay is that they reproduce in a first year and can release 2 or 3 or maybe even 4 batches of eggs that year, and potentially even the same in a second year after that. So they're, usually each female is producing a lot of eggs, and each one of those birds of eggs can be something like 2 to 3 million eggs. So she could potentially produce 10 million or more eggs over her lifetime. So it may be one mating event, but each female that survives to the end of her life is producing a whole lot of eggs. So yeah, great question.
And a follow-up on the blue crab research, is the Smithsonian partnering with other local organizations on this?
Yeah, so we partner in a couple different ways. One is that for these tagging events, we've tagged something like 50 to 60,000 crabs over the last 20 years. That's a lot of crabs to catch and tag. So when we do that, we are usually partnering with local commercial fishermen who are really good at catching a lot of crabs, and we usually have one person on the boat with them helping to sort of package the crabs up in a way that's really safe for the crabs, and then transfer them over to one of our boats where we have a team of people on the back of the boat attaching tags and putting the crabs back in the water. And the other way that we partner is that we work really closely with the state management agencies in Maryland and Virginia to share the data once we get it, so that they can incorporate our data into the management as well. And finally, the key group to make all of this work is that we only get tags back that fishermen call in. So we've worked with hundreds now of different commercial fishermen and recreational crabbers who might be going out on their boat or their dock who are catching crabs and calling that information back into us.
That's great. Jared, this might be a good one for you. We have a question if the Smithsonian still has bison like we did at the turn of the century.
Yeah, good question. Yeah, I mean, it was the first species that was brought to the National Zoo on the Mall over a century ago. We do have a few bison that act as ambassadors that are still at the National Zoo. But importantly, we're also involved with the project in Montana focused on bison and restoring bison with the American Prairie Reserve. So the American Prairie Reserve is establishing or trying to establish the largest protected area in the continental United States, with bison as the flagship species and really trying to facilitate movement across this landscape as well. So yeah, so it's still really an important species for the Smithsonian.
Wonderful. And another question for you. How hopeful are you about the plight of giraffes and Asian elephants?
Well, I can say working with our team, it's really encouraging. I think there's a lot of people that are doing everything they can to save both species. And in all these cases, working with local people. And I think the most important thing is to see people on the ground that are looking for our assistance to restore these animals to their habitat. So I guess I am optimistic about, conserving and saving these species into the future.
Great, I'm glad to hear that. So one question for everyone, how is climate change affecting your work and species migration in general?
Who wants to go first?
I can go first. One of the things that we certainly are thinking about a lot is climate change, and for things in the water, there, animals living in the water. There's two sort of key components to that. One is that the water is warming up, and the second is that as water warms up, it holds less oxygen. So warmer water makes it harder for animals to breathe, to get enough oxygen when they're sort of breathing underwater. So those are two of the things that we think a lot about. But with the migratory fish, for example, we think they're probably showing up earlier and earlier each spring. So we have a new project this year where we're deploying temperature sensors with a postdoctoral fellow named Henry Leggett, who's deploying these temperature sensors and going to be modeling in really fine scale how stream temperature changes during the spring and how climate change might affect that. And then in the ocean with sharks and rays, what we're expecting to see is that their northward migration in summer is going to take them even further north. And the species we're working on mostly live in the mid-Atlantic and stay south of Cape Cod. But the climate predictions suggest that as waters continue to warm in the summer, they'll be able to move further north into the Gulf of Maine, off the coast of Maine, where there aren't as many large predators like that. And so it could really change the way the ecosystem and fisheries work there. And so that's one of the topics we're really interested in studying with those species.
Yeah, for birds, similar, they're showing studies where the timing is off, arrivals happening earlier. And also they're seeing, which is interesting, and a lot of research is just starting here with range shifts. So birds are either not migrating as far north or staying resident all year instead of choosing to migrate. So it's changing the distribution of some species. For the herons, we're just starting to look at this information with the tracking data, and especially for birds that are wintering on Florida Everglades and Cuba. those areas are predicted to have more droughts or, stronger storm systems that could inundate saltwater or the freshwater with saltwater. And these can all affect foraging and roosting sites and foraging sites for birds. So.
Yeah, and I guess I would just say for terrestrial mammals, I think the complexity is that many of these animals are constrained to protected areas. And a lot of these protected areas right on their boundaries is where you have lots of human development. And so from a climate change perspective, the problem is, as the climate changes, do these boundaries allow animals then to shift with the changes and actually track that vegetation? And that's certainly what our concern is in the system in East Africa with the wildebeest. In East Africa, it's expected to get hotter, but also wetter. And so in some ways you could say, oh, maybe vegetation conditions might be better for the animal, but the increased heat means that there's less water actually reaching the surface. And so there's complex interactions occurring on the landscapes or across the world.
Yeah, that all is unfortunate, but definitely makes sense. For you, Amy, what is the impact of wind farms on migratory birds?
So I'm not an expert in this field, but what I do know is maybe about 10 or 15 years ago, there was a lot of surveys starting on wind farms to look at the impacts on migrating birds. Because when birds migrate, they want, especially raptors or larger birds, they often are using migration routes where people want to put wind farms because it eases their migration and they can coast on the wind. And that's where wind farms want to go. But a few years back, they did a lot of surveys and we're starting to retrofit windmills to be more bird friendly, I guess, with longer blades. So windmills used to have shorter blades and spun faster. So now they'll have longer blades and they spin slower. Wind farms might work with local biologists when they turn on their windmills. And so some have like a schedule, which, birds are always migrating, but at least it can reduce some impacts. Or if more birds are migrating at night or during the day, then they might turn off the windmills during those times. But yeah, it is a issue that a lot of people are researching now, and some data has come out. So maybe Smithsonian will get involved in some of these projects sometime and have a better answer for you.
Thank you. Matt, so a question about dam removal in fish migration. If a dam has been in place for many years and is removed, assumedly the generations of fish would have never known about it. Why do you think the first few generations of fish going upstream would even now venture that far?
Yeah, that's an excellent question. And that's, it's one of the things that I'm really excited to see this year. I mean, this is exactly the time of year when these fish are migrating, and we should have an answer really soon for this species in this place. But people have seen studying other dam removals in other places with salmon on the West Coast, for example, that when a dam is removed, they move upstream. And we've seen that other places with the species we're working on in the Chesapeake. The Rappahannock River is an example, had a large dam that was removed about 15 years ago. And we know the fish are moving somewhat up past it. But in the Patapsco River where we're working, we see fish that, that migrate upstream and just hang out at the base of the dam, trying to go further upstream. And then I think, in the past, they've probably ended up reproducing there and then going back out to the ocean. But, our guess is that those fish that come up and are blocked by the dam itself will probably migrate further upstream as well. But, we don't know. I'll check back in a month or two and we'll tell you the answer.
Excellent. We'll stay tuned. For you, Jared, so this donor said everyone loves large mammals like elephants and giraffes. What do we need to know about smaller species that are also important in the overall picture?
Yeah, well, that's a great question. Obviously, some of the larger mammals are a bit easier to track, right? And maybe they garner a little bit more attention. Part of the reason I study large mammals is because they need large habitat areas. And so presumably by protecting the large animals, you're protecting some of the smaller ones at the same time. But all these species are individuals. They all have individual habits. And so I think combining what we know from large mammals to small mammals gives us a better assessment of what's going on in the system. Certainly when I spoke about the wildebeest across the system, you know, we're not just interested in wildebeest. We're interested in kind of the whole suite of species across that landscape, so that when we are prioritizing activities across the region, it doesn't just conserve one species, but hopefully it's amenable to the whole community of species that are trying to move across the landscape. So yeah, I think it's a merge of everything all into one. It's a good question.
Thank you. And this will just be our last question, just because we're running short on time. What do you think is the most helpful thing that we as individuals can do to help preserve biodiversity?
I'll start. Oh, yeah. Sorry. You
Go
want to go? That's a great question, and thank you for bringing that up in this discussion. There's a new campaign that Smithsonian's a part of in the Migratory Bird Center, and it's called 7 Simple Actions, and it's how to be bird friendly. And it is on the Migratory Bird Center website, or you can Google it. But some important things we can do are plant native plants in your yard, keep cats indoors, try to buy bird-friendly products or things like that. Coming to these talks is really great in learning about what we do and how, just get us thinking about connections between species and learning about tracking animals. And so this is awesome that you all have attended. Visit the zoo if you live in the area, or if you plan to visit, that's also a great way. So I'll stop now, but yeah, if you want, you can Google 7 simple actions to be bird friendly, and there's a lot on that website.
Yeah, and I guess part of my answer, I think, overlaps directly with Amy's, and I think, we protect things that we really care about. And so obviously I talked about international species and large mammal conservation, but even just getting really interested in the birds in your backyard, I think is a really good way to say, we really want these animals in the future.
And I would just add, sort of on the fisheries side, a great thing you can do is learn about which fisheries are sustainable, what types of seafood are sustainable and which ones aren't, and try to focus on consuming sustainable seafood. There's, despite what you hear sometimes in the news, there are fisheries that are very sustainably managed, and so that's a great way to help with conservation of biodiversity in the oceans.
Wonderful. Well, thank you all for those thoughtful answers. And I just want to thank you again for being here with us today and sharing your expertise on the movement of life and how this vital inter-unit initiative is to understanding and conserving biodiversity across the globe. I'd also like to invite everyone to join us next week for a special Earth Day lecture with the Smithsonian Environmental Research Center focused on ecology. The title of that program is Earth Optimism Panel, The Hidden Workings of the Forests and Their Future. And we'll be sharing the link to register for this program in our post-event email, so please be on the lookout for that. Thank you so much for joining us today and for your invaluable support as a Friends of the Smithsonian member. We look forward to learning more with you soon. Thank you.